Hello students, welcome to the theory class of Principles of Plant Breeding. Today's topic is on plant breeding methods which can be used for both self and cross-pollinated crop plants. We'll discuss on hybridization techniques and backcross breeding method. So by the end of this class you'll be able to answer what are the breeding objectives of hybridization, why backcross method is used, what is recurrent parent and non-recurrent parent, and you'll be able to understand how to develop a hybrid variety, how to transfer dominant or recessive genes using backcross method. So before we start, let's discuss some of the important concepts of related to the topic. First of all, the difference between, let's compare the difference between pure line and a hybrid inbred. Okay, so what is pure line and inbred? If we look into the similarities of these two lines, okay, they are produced by selfing, okay, continuous inbreeding. Both these lines are produced by continuous inbreeding, okay, and in both the case, in the resultant will be homozygous, okay. And coming to difference, okay, we have here, it is a pure line is a homozygous plant developed from self-pollinated crop plants, and inbred is a homozygous plant developed from cross-pollinated crop plants. Okay, well, the difference lies here that by under natural conditions, cross-pollinated crop plants are habituated for cross-pollination, open pollination, while in case of self-pollinated crop plants, they are habituated for, they are accustomed for selfing. Okay, so there is a problem when there is a selfing in cross-pollinated crop plants. There is a problem in growth and development, okay, and vigor of the plant, which is called as inbreeding depression. Okay, if we self this the same pollen into the same stigma of the same flower or same plant, fertilization of flowers of the same plant, it will produce inbreeding depression because they are not habituated with it. So although we produce it because we need homozygous plant as a parent in hybridization program. Okay, so we'll be discussing inbred, we'll be using this term inbred in hybridization program. So it is important here, important to know the concept of inbred lines. And coming to a comparison between a test cross and a back cross, okay, quite similar terms. And in the first case, test cross is a cross between a dominant phenotype with a recessive phenotype. Okay, say for example, when we have a two parents, capital A and capital A small a, crossing which will form a heterozygote capital A small a. Okay, so if we cross capital A small a to small a, which is a recessive phenotype, so it will produce it will be called as test cross. Okay, usually this is used to identify whether the progeny is a homozygote or a heterozygote. Okay, so this is significant in testing of a progeny. Okay, while in case of backcross breeding program, it is a cross of the F1 with either of the parents. Okay, and this is usually not a test, this is a method to transfer genes from the a donor parent to a recurrent parent or recipient parent. Okay, coming to the hybridization program, some of the concepts of hybridization technique is well, hybridization is basically crossing of two plants, usually dissimilar plants or diverse, you can say. And in this case, it is a process of usually placing of a pollen grain of one genotype in, usually called as male parent, into or onto a stigma of another parent called flower of another parent called female parent. Okay, so when they are crossed, the resultant will be called as F1 hybrid. Okay, and this is not so in F1 in hybridization program. We usually go for two different sections. First one is production of F1 hybrid. Okay, if it is yielding huge amount of if it contributes to huge amount of heterosis or hybrid vigor, we can directly release as a variety, or we can further go for F2 and further generations and use other breeding methods such as backcross breeding methods. Okay, so in this case the F2 and F3 and further generations are called segregating generations. These are also significant, which plays a great role in plant breeding program. Coming to the objectives of hybridization. So why we are doing hybridization? The first and foremost aim is to transfer one or few qualitative genes, that means oligogenic genes, easily identifiable, heritable and controllable genes from one parent to another. Okay, one parent to another or one parent to the progeny. Okay, and next is to improve one or more quantitative character. Although we have already the transfer of qualitative character is quite easy, improvement of one or more quantitative character, that means character polygenic character governed by several minor genes in cumulative action. Okay, so in this case we have to introduce several parents. Okay, we have to accumulate genes from several parents and into a particular uh, hybrid variety. Okay, so this will be the broad perspective of it. And number third is F1 progeny, development of F1 progeny or F1 hybrid variety. Okay, coming to the first one, we have combination breeding. Well, combination breeding is also known as conversion breeding. In this case it uses parents, transfer of significant genes from several parents, one or few parents or several parents into a uh, particular progeny. Okay, so in this case uh, collection of superior plants which have separate superior agronomic characters. Okay, some of them may be disease resistance, some of them may be abiotic resistance, some of them may be fruit quality, or some of them are having higher tiller number. Okay, accumulating all these significant character into one uh, variety is called combination breeding. Okay, so what is yield? Actually it is a quantitative character. Okay, so in quantitative character, so to obtain a huge amount of quantitative character, it can be contributed by transferring genes governing the tiller number, grain per spike, taste, weight, disease resistance. Accumulating all these genes from different parents would be a good perspective. Okay, and coming to transgressive breeding, different type of breeding here, which is a crossing of two different parents or diverse parents forming F1. And uh, not only forming F1, if we self F1 generation, it will produce F2 and F3 generations. Okay, so uh, when we have an F1 superior to either of the parent or both the parent, it is called as hybrid vigor or heterosis. But when we have in a heterogeneous F2 population, if we have any superior progenies which surpasses or which outperforms the parents, it will be called as transgressive segregants. Okay, selection of these genotypes from these transgressive segregants, it is another breeding perspective, and it is called as transgressive breeding. Okay, again transgressive breeding is selection of superior progenies from F2, F3, F4 generations. Okay, not from F1. Okay, so this is again another breeding method. Again, coming to hybrid varieties. Okay, so in this case it is the uh, superiority of F1 over their parents. Okay, so it is based on heterosis. In so usually in case of self-pollinated crop plants, F1 is more vigorous and higher yielding than their parents. Okay, and as you can see here, when there is a cross between two parents, okay, the F1 hybrids are uh, usually tend to give higher yield than their parents. Okay, so it may be contributing from the heterozygosity that they used to possess. Okay, we have three concepts of heterosis: dominance hypothesis, overdominance hypothesis, and uh, epistasis. Okay, so in overdominance hypothesis, it is due to the heterozygosity. Okay, although both these parents doesn't have the vegetation the f1 is having right now. Okay, so the contribution of the vegetation, large vegetative growth, is maybe because of the heterozygosity, and and of course contribution or accumulation of both the characters. Okay, and coming to the types of hybridization. Okay, uh, it can be classified into two: intervarietal hybridization and distant hybridization. Okay, so in when it comes to uh, popularity, okay, or most commonly used hybridization program, it is intervarietal hybridization program, because in most of the cases we usually go for hybridization between two different varieties because of the compatibility and because of the handy in which we can work on. Or otherwise, if we want to cross between two different species, it will be challenging. It needs high-end equipment. It needs high-end equipment such as biotechnological tools, which has to be assisted in usually when we go for distant hybridization. So coming to the second category of um, hybridization, which is distant hybridization. So usually in case of intergeneric hybridization, it is assisted by biotechnological tools. Okay, say for example, plant tissue culture, such as somatic hybridization. Okay, fusion of two different cells, protoplast or hybrids. Okay, and then it can also be assisted by uh, transgenics. Okay, and in this case, interspecific hybridization, it can also be assisted by uh, plant tissue culture methods. Okay, ovule culture, okay, or embryo culture, embryo rescue, and many others. Okay, so in this interspecific hybridization, the compatibility is uh, the incompatibility between the parent is lesser compared to the intergeneric hybridization. Okay, there will be a better compatibility between parents. Okay, and there are successful progenies between when crossed with two different parents. Okay, although there is a rare chance, but there are successful cases where interspecific crosses can be made without plant tissue culture. Okay, so let's see one by one what are these. Intervarietal hybridization, although this is also known as intraspecific hybridization within the species. Okay, and in this case there will be cross between or there will be hybridization between same species and two different strains, or you can call it varieties or races of the same species. Okay, and in crop improvement program, intervarietal hybridization is the most commonly used hybridization methods. Okay, and coming to uh, according to the complexity of the parents used. Okay, the crossing or hybridization can be classified into simple, okay, which uses two parents forming an f1. Okay, so this is the simplest form, and complex cross. Okay, so in complex cross there are more number of forms. There is flexibility and you can use many number of parents uh, according to the convenience and according to the need of the plant breeder. Okay, so when there is a three-way cross, so it's called conversion. It is also called as conversion cross. Okay, because we are going to transfer the genes from collectively from A, B, and C. Okay, if we are using these three parents in three-way cross. Okay, but in case of three-way cross, first of all there will be crossing between two parents A and B, which forms the cross AB, and there will be the F1 will be crossed with the third parent C. Okay, so this usually is done during during cases such as incompatibility of parents. Okay, if A is compatible with B and C is compatible with B, so when we have a cross between A B, okay, the homologous chromosome of B can pair with C. So that's why the incompatibility between A and C is uh, overcome by having three-way cross. Okay, so this is another uh, way of making cross between incompatible parents. And okay, examples of another complex cross using four parents. Okay, so not only three parents, we also use four parents, which is also called as double cross hybrid. Example would be in maize. Okay, and so in this case also uh, various various traits are accumulated. Significant traits or agronomically superior traits uh, are accumulated in a particular uh, variety. Okay, so in this case the trait not only concerns with qualitative traits. Okay, the trait here we are discussing is uh, it can be quantitative traits here. Okay, here again a more complex, again more sophisticated cross, which is called as which is includes 8 parents. Okay, and next is distant hybridization. So what is distant hybridization? Crossing between two different species, uh, plants belonging to two different species or two different genera. Okay, so it is usually called as interspecific or intergenera. Okay, so according to if the two plants, the two parents which are going to cross are from two different species, it will be called as interspecific, and if it is from two different genera, it will be called as intergeneric. Okay, and coming to, so after distant hybridization we have hybridization procedure for self-pollinated crop plants. That means for those plants where there is complete flower, okay, and presence of androecium and gynoecium, or we can call it as hermaphrodite flower. So in this case the first thing to do is choice of parents, better performing parents or disease resistance parents or insect resistance parents depending on the need of the plant breeder. So the parent will be chosen, and after that uh, evaluation of the parents based on the progeny, because it may be a false alarm, because the character that you uh, saw in the field and what you have selected maybe because of the environment effect. So you have to grow the progeny, okay, and find out the real or genuine character, okay, and based on the progeny performance. Okay, so after the progenies are evaluated and the characters are checked, so we have to emasculate the parents. What is emasculation? It is the process of removal of the androecium or anther from uh, the hermaphrodite flower, making it a female flower, okay, a pistillate flower. So the staminate will be removed, and the stamens will be removed, and the flower will remain as pistillate. Okay, complete pistillate. So what is the process? Usually it is done with a forceps, and in that case for minute flowers, such as in case of pulses, we usually pull out the keel petal. So in pulses the keel petal is attached with anthers, so there is no need of removing separate anthers for that. Once the keel petal is removed, so the stigmatic surface will get exposed. Okay, and and things should be taken care that the exposed stigmatic surface is not pollinated by foreign pollens, unwanted foreign pollens. Okay, and for this you can use bag tags or straw, okay, straw pipe, in order to avoid the foreign pollens to contaminate the stigmatic surface. Okay, and in this process, what is the timing for emasculation? Timing of emasculation is very important. Why? Because if it is going to be done in the evening. Okay, in the evening when sun goes down. Okay, and usually at 4 to 5. Okay, and why is this important? Because if we do emasculation in the morning, okay, right after the petal is removed, it is exposed to sunlight. When it comes to 10 o'clock to 11 o'clock, it will be exposed to sunlight, and it will start withering. Okay, this portion will start withering, and this will get the moisture content inside it will get exhausted, and it may die. Okay, and because of that, in order to avoid this, we do emasculation in the evening and pollination in the next morning. Okay, so crossing or pollination is done usually at 6 o'clock in the morning to 9 o'clock uh, in the morning itself. Okay, so and once the so pollination is done once the pollination is done, you have to make sure that the bagging is done, and label is done, and in the label you have to write date of pollination, date of emasculation, and time of emasculation, such as parents. Okay, parents of the cross. Okay, and harvesting right after bagging and leveling, you have to wait for the seed filling, seed maturity, and you have to harvest the F1 seed, and you can raise F1 generation. Next thing is when you're supposed to grow, or when you're supposed to produce commercial hybrid seeds. Okay, instead of hybrid F1 hybrid. So this is this method of hand emasculation and pollination. Sometimes it is used in private companies, but it is not usual. When there is another mechanism for hybrid seed production, say for example in those crop plants where male sterility is present, so we usually go for hybrid seed production using male sterility, because it is much easier, and because emasculation step is avoided, which is a tedious process and it requires skills. Okay, so the cost of production of seed is quite high if it involves huge skill labors. Okay, so in this, what is commercial hybrid seed production? It is the use of either genetic male sterility, cytoplasmic male sterility, or cytoplasmic genetic male sterility system to produce huge amount of seeds. Okay, so in this process, the most important thing is the male sterile line, which is called as A line. It has to be crossed with a maintainer line called B. So you have seen the genetic constitution, recessive, homozygous. When crossed with heterozygous, it will produce the same individuals in the same ratio. Okay, we are going to avoid this. Okay, and we are going to utilize this as a continuous supply of female parent, or also called as your male sterile line. Okay, this is required. Why required? Because the more number of female line, the more will be the number of seeds. Okay, although the pollinators is also desired parent, the supply of pollen is also necessary. So since there is no need of since there is no need of emasculation and high-end techniques in this pollinator parent, we are going to grow it in a field plot, and usually in certain cases the number of pollen grains is usually higher than the female eggs. Okay, or so that's why it is important that we need a huge amount of or continuous supply of the male sterile line. Okay, uh, and the pollinator parent, when it is crossed, it will produce the hybrid. Okay, so this process of production of male sterile line is called male sterile line production line, while in this is case the process of crossing of male sterile line with the pollinator parent is called hybridization, and the product is your hybrid F1 hybrid. Okay, and coming to the hybrid seed production in cross-pollinated crop plants, it involves three steps. Okay, first of all, choosing of the parent, which is called, or choosing of the parent or development of the parent, which is called inbred line. Okay, well, we have already discussed earlier what is inbred, and once the desirable inbreds are developed, are chosen, so we have to evaluate the inbreds. For what? For their superior performance and their combining ability. And what are the combining ability that we have studied? General combining ability and specific combining ability. Okay, when these three things are fulfilled, we are going to use them in hybrid seed production program. Okay, let's say one by one. So in the step one is for production of inbred lines. So the base population, from the base population we are going to subject the lines, desirable lines for with continuous breeding or inbreeding to produce inbred lines. Okay, continuous selfing or continuous inbreeding will induce the open pollinated cross fertilized plants into homozygous plants. Okay, so once the homozygosity is attained, so we have to evaluate the phenotypic performance in replicated yield trials first of all, and we are going to select those which are superior based on the replicated yield trial. Okay, and after a few lines are selected, some of the lines are selected, based on that we are going to evaluate the top cross test general combining ability. What is general combining ability? How are we going to evaluate the general combining ability? Say for example, we are going to evaluate the GCA for A. We are going to cross the parent A, the inbred A with several others. Okay, or also called, this is also called as open pollinated variety. So when it is crossed, they will give several progenies. Okay, we have to evaluate the progeny performance, average progeny performance. Okay, if it is higher, then it will be called as high GCA. Okay, and the next thing is single cross test specific combining ability. So for specific combining ability, it will be if A is high and if the general combining ability for A is high and B is also high, we have to go for the SCA test for B cross A and A cross B. Okay, so performance based on A cross B and B cross A will determine the specific combining ability. And what you have to remember here is, for hybrid seed production program, for F1 hybrid seed production program, estimation of both GCA and SCA is required, while in case of synthetic seed production program, synthetic variety production program, we need only GCA evaluation of the component lines. Okay, so once these three are evaluated, phenotypic evaluation, performance evaluation, GCA and SCA evaluated, so the inbred lines are going to composite and form several superior inbred lines with high combining ability. Okay, once they are formed, so we are going to use this for production of single cross or double cross hybrids. Okay, so this is done, hybrid is produced. Next thing is a requirement of a backcross program. Okay, so next thing is backcross breeding program. So what is a backcross? We have already discussed in the first, we have already introduced backcross in the first slide, and it is the process of crossing between two parents. What kind of parents? One of them is called recurrent parent, which has all the agronomic character except one or two character. Okay, desirable character which the breeder is wishing to transfer from donor parent. Okay, and another parent called donor parent, where the agronomic characters are lacking but they have the desirable one or two character. Okay, so our intention is to transfer one or two character from the donor parent to the agronomically superior parent, high yielding parent. Okay, say for example disease resistance introduction. So backcross breeding program is usually done for transfer of characters with high heritability and which are governed by one or few genes. Okay, and sufficient number of backcross breeding program is necessary. Why? Because once the transfer is done, these genes which are transferred from donor parent is usually associated with genetic load. What is genetic load? Some desired genes, some undesirable genes are associated with the desired genes transferred from donor parent. So in order to avoid this, cut down these undesirable genes, we need enough amount of backcrosses with the recurrent parent. Okay, again and again. So coming to one of the module of transfer of dominant genes, okay, which is the simplest one. First of all, we have as usual non-recurrent parent, okay, non-recurrent parent, or called as donor parent, and then we have recurrent parent, okay, variety A. Okay, so in this case we'll be transferring the rust resistance gene to a susceptible line. Okay, so in this case recurrent parent will be agronomically superior variety, that means high yielding variety, but which is lacking in one character, which is resistance to rust character. Okay, and in this case this is a donor parent which doesn't have high yield, doesn't have high agronomic character, but having rust resistance character. So our intention is to transfer these dominant genes governing rust resistance to the progeny. Okay, so in this, first of all we'll go for hybridization. Okay, hybridization will lead to 50% of this genotype and 50% of this genotype, contribution of both. 50% of recurrent parent is retained. Okay, so we cannot release the hybrid as such because there are lots of most of the genes transferred from the donor parent is not beneficial. Okay, because they are not contributing to agronomic high yielding. Okay, so in fact it will lead to decrease in yield, okay, in the F1. So what we will do, we'll retain regain the rest of the recurrent parents genome. Okay, so how to do it, we have to backcross again and again to retain the amount of uh, genetic losses during hybridization. Okay, so once F1 is done, 50% of the recurrent parent is there in F1, and when we do so, we are going to cross again, the F1 is going to cross again with the recurrent parent. In the BC1 will get how much? 50% plus 50% of the 50% remain here. Okay, so in every, what we have to remember is in every backcross breeding program, 50% of the recurrent parent is regained in the 50% of the backcross breeding recurrent parent is retained in the backcross generation. Okay, so in every generations of backcross there will be retention of 50% of the recurrent parent. Okay, so 50% plus 25%, that is 50% of 50%. Okay, which is total 75%. In this case, okay, when we cross between capital R smaller and smaller, there will be the progenies will be 50% smaller and capital R smaller. So the progenies smaller, which is rounded by blue, will be rejected. Okay, because we are not going to use it, and the heterozygous will be used in the backcross breeding program. Okay, so time and again, if we backcross, there will be retention of 50% of each. So next, in the next round of backcross breeding program, there will be 50 plus 25 plus 12.5, which is 50% of 25% remaining recurrent parent. Okay, which is now 87.5% of recurrent parent. Okay, and next in BC3 generations, 93.75% of recurrent parent, and then in BC4 there will be 98.7% of recurrent parent. So until and unless you have our intention would be to reduce as much amount of the donor parent's genome, because we don't want deleterious gene or undesirable gene from the donor parent to attach along with the desired gene. Okay, so we want as much as recurrent parent along with only the genes, okay, Okay, only the donor genes desire conferring the disease resistance. So at BC6 you will witness 99.7% of recurrent parent, which would be desirable. Okay, although there will be presence of genetic load there also. Coming to the transfer of recessive genes, there will be a slight complexity here, because in this case same thing here. The only difference is the donor parent, the desirable gene is governed by recessive gene smaller, while uh, the the recipient gene is capital R. Okay, we have two parents, okay, as usual, recurrent parent and non-recurrent parent, also known as donor parent variety B, and recurrent parent as variety A. Rust resistant genes governed by smaller and susceptible by capital R. Okay, we are going to transfer this gene to susceptible lines. Once F1 is there, 50% of the recurrent parent is retained. So when it is fertilized, it will produce these two lines, isn't it? And then when they are self, okay, it will produce these progenies. How? If we self this, selfing doesn't mean we are going to cross between these two, okay, that we are going to self this capital R with capital R, it will produce capital R, okay. And if there is crossing of capital R small r with capital R small r, there will be production of 1:2:1 ratio of capital R small r and small r, okay. So in this we are going to have these type of genotypes, okay, and we are going to avoid this because we will be needing only recessive homozygous genotype. So the self BC1 will be called as BC1 F2 generation because it shows segregation. So when the recessive undiscarded recessive homozygous kept smaller is crossed with the susceptible line, it will then produce the heterozygous type, okay. And when the heterozygous capital R smaller is crossed with capital R, it will produce BC3 generation, okay. And in BC3 generation, this capital R, when self, it will produce again the same thing in this. So we are going to discard here these two and take this, okay, takes smaller and go on, and the cycle continues as long as the amount of genetic load is reduced as much as possible, okay, 99.7 most of the time. So the process of transfer of recessive gene is associated with a little bit complexity when compared to transfer of dominant genes, because it involves selfing, a generation of selfing, while it is not there in dominant gene transfer. Now coming to merits of some of the merits of backcross method, okay, the genotype of new variety is nearly identical with that of the recurrent parent except for the gene transfer, okay. The new variety that is produced is nearly identical with that of the previous variety, okay, and there is uh, no loss of genetic variation, okay, or no loss of agronomic character. So recurrent parent will retain its yielding character, okay, most of the uh, superior character added with the desirable character, okay. Much smaller population is needed in this method when compared to pedigree mix, because we are going to use only two desirable parents and not more than that, okay. We are not going to produce huge amount of F2 generations also, and this is the only method for interspecific gene transfer and for transfer of cytoplasm, okay. So though in conventional breeding program it is the method to transfer in and hybridization and transfer of cytoplasm, okay, and also male sterility. Susceptibility is in a well adapted variety can be removed without affecting its performance and adaptability, okay. So these resistance can be introduced easily, and this is usually possible with qualitative genes, okay. Coming to demerits of backcross breeding program, the new variety is generally and generally cannot be superior to the recurrent parent, except for the new variety is not uh, far more superior. Okay, after doing several backcross breeding program, the new variety is not far more superior. Only improvement is the donor genes. Okay, so this our intention is this only, but the thing is, uh, in uh, in transgressive segregation, in transgressive breeding, or you can say pedigree method, you can expect much more. Okay, because of the heterozygosity or the various genetic combinations between the diverse parents. Okay, because in case of pedigree method, the two parents are both desirable, while in case of backcross breeding program, only one is desirable and another one is not undesirable. Okay, only having significant character. So there won't be chances of much improvement except for the gene in transfer. Linkage drag, which is close association of the undesirable genes along with the desirable genes, which is transferred in the backcross. Okay, and then hybridization required for each backcross. Okay, hybridization, or that means backcrossing is required in every generation, which is difficult, time taking and costly. And in case of transfer of recessive genes, there is selfing generation also. Coming to some of the achievements of backcross breeding program, rust resistant genes has been transferred to Kalyan Sona from several diverse sources such as robin, HS19, bluebird, etc. TIF23A of Bar T23A was used in Backcross program with resistance lines from India and Africa to develop downy mildew resistance Milstein line, such as MS521, MS541, etc. And Gossypium herbaceum varieties, Vijalpa, Vijay, Digvijay, Kalyan are some of the cotton varieties developed by backcross breeding method. So now coming to the end of the slide, if you have any questions, please do ask.